IB Biology Topic 3.2 Chromosomes Point 2. Some prokaryotes also have plasmids, but eukaryotes do not. Here we'll be looking at what a plasmid is, and why some prokaryotes have them, but why no eukaryotes do. First we'll be looking at the definition of a plasmid. So a plasmid is a small, extra DNA molecule that is commonly found in prokaryotes, but very unusual in eukaryotes. These plasmids are usually small, circular, and naked. And they contain a few genes that may be useful but that are not necessary for life. And here life is the basic life processes. And also I'll add that here naked, if you remember from the previous video, means that it is not associated with proteins. I've also added down here one of the example uses for a plasmid. And in this case, it's that a plasmid can have the genes that are needed for antibiotic resistance. Plasmids can also have carry genes for other kinds of resistances or other uses, and we'll see those near the end of this video. Our next point is that plasmids are not always replicated at the same time and rate as the chromosome. There may be many copies of a plasmid in a cell, but plasmids may not be passed down to both daughter cells. On the other hand, plasmids may be transferred from one cell to another, and they can also cross to other species. I've added an example here. So a cell dies and exposed plasmids are absorbed by other cells. In this case, you have to imagine that certain parts of a cell are useful to other cells as well. In this case, the part that is useful is a cell's plasmid. For example, a plasmid can allow a cell to survive in very high or very low temperatures that other cells may not. So here I've drawn out a certain example. First we have a cell with a plasmid that allows for cold resistance, and I've just written plus cold. Now if this cell were to leave its cold climate and travel down to where there's a normal climate for other cells, or rather where other cells can live, if this cell dies in this normal climate, and another cell absorbs this plasmid that allows for the cold resistance, then this other cell can move to this cold climate and function normally. And this kind of method of transferring genes is called the natural method of gene transfer between species. This is the method of transfer when one species provides another species with some genetic information, in this case a plasmid, without replicating or producing this information for it to be passed down, for example, to daughter cells. And in this case, we know that plasmids are not passed down to the daughter cells of the cell. This method of gene transfer is also used by biologists to transfer genes between species artificially. And this previous example would work as well. So if a biologist took this plasmid that gives cold resistance from a cell and gave it to another cell, they could reproduce this effect and allow this second cell to survive in the colder climate without the second cell needing to absorb the plasmid from the first. So in this case, the biologist would inject or in some other way give this plasmid to the second cell without the second cell ever needing to come in contact with the plasmid from the first cell, or rather without having to absorb it. And this can also be reproduced on a larger scale. So maybe a biologist will be able to artificially, or rather synthetically, create the plasmid and give the plasmid to many cells to either test and see how the cell will function, and in this case try to improve the plasmid, or maybe just test the plasmid to see whether the synthetically produced plasmid maybe has some kinds of consequences for the cell, and the cell actually can't survive because there are some issues. This final slide is the PLW1043 plasmid. And here we can see the shape of the plasmid, as well as how certain parts are associated with certain abilities. And here we have 4 kinds of resistances, as well as this part of the plasmid that has genes to help the plasmid spread. Each of these individual traits can be taken out of the plasmid, for example, by biologists, and then synthesized and added to other cells to allow them to have one of these abilities. And through research and testing, we're also able to synthesize certain traits and to combine either resistances or combine beneficial abilities, not just here for allowing the plasmid to spread, but for example, allowing the cell to move quickly or to need less food, be able to absorb the food faster, and other kinds of abilities that will help it to outcompete all of the other cells that are in its environment.